Hi, so I'm going to walk you through the premise of one of these experimental designs. This particular one is looking at the effect of cranberry supplements. So I have some azo cranberry supplements on urine in regards to its activity against E. coli. So E. coli is a major cause of urinary tract infections. Many people, especially women, know or have heard that if you drink cranberry or take cranberry supplements, it can at least help with the pain of a urinary tract infection and maybe actually help treat the urinary tract infection. So this research project aims to ask this question. Does azo affect the urine's ability to act as an antimicrobial agent against E. coli? So what I have here is I have a urine sample that was taken yesterday by me prior to taking any azo. And then I took a dose of azo last night and then another one this morning. And then I just took a second urine sample a little bit ago, which gives me about 4 hours after I took my last dose of azo. That should be enough time to make it into the urine. And what we're going to do is we're going to combine the urine samples with E. coli broth. I also am going to combine some of that E. coli with just regular nutrient broth, which E. coli grows really well in. And what I'm going to do is I'm going to look to see if there is a difference in terms of how E. coli grows in each treatment. So one treatment will be E. coli plus nutrient broth. Another treatment will be E. coli and urine prior to taking azo tabs, and then a third treatment will be E. coli and urine after taking 2 doses of azo. All right, let's get started. Okay, so using my wax pencil, I'm going to start by labeling 3 test tubes. I have one that is control, one that is urine prior to taking azo tabs, and one that is urine after the azo tabs. All right, to each of these, I'm going to add about half a milliliter of E. coli. Now I'm going to use the same broth for each sample. And the reason why I'm picking the same broth is so that I can ensure that the starting concentrations are pretty consistent. Mix it up a little bit first. Okay, to my control, I'm going to add 1.5 milliliters of nutrient broth. Again, E. coli should grow really well in this, so that will give us a good baseline for how well E. coli is going to grow. And I apologize, I misspoke a little earlier. I did 2 mils. That'll give me a total of 2.5 mils, which will allow me to take 1 mil from 2 different time points out of here with a little bit left over. So I did 2 mils, so that means that to keep it consistent, I need 2 mils of the appropriate urine as well. So we change our tips to prevent cross-contamination. And let's see. Okay, so this is my urine sample prior to ingesting any azotabs. Okay, so we'll do 2 mils of this as well. So again, the total amount in here is 2.5 mils. And finally, the urine after azotabs. Now, ideally, I would have taken a first morning urine sample in both cases, but because we were given, the faculty were given very short notice that we had to be off campus sooner rather than later, I was not able to plan or give myself enough time to collect samples first morning prior and first morning after. So I collected my urine yesterday afternoon, and then I collected my urine again, not my first morning urine after the azotabs, but around 11 o'clock. So there are some slight differences in the color of these urine samples. The pre-urine sample is a little more dehydrated, and the post-urine sample is a little more dilute. So that may affect the results, and unfortunately it's a type of introduced bias that I don't know how to correct for given the constraints. So it wasn't the ideal setup, but it is a setup that I tried to reduce as much noise as possible. So for instance, I drink coffee, and I tried to make sure that my food, the time after drinking a cup of coffee was pretty consistent so that both of them had the same amount of caffeine in the urine if that was going to impact it. I tried to keep some other stuff consistent as well, but obviously it wasn't perfect, as evidenced by the fact that I've actually had 2 different colors, suggesting at least my levels of hydration were different. Okay. So now what we have our urine samples mixed and our nutrient broth mixed with E. coli. And so my next step is going to be to do serial dilutions. Now what I want to do is a set of serial dilutions of these samples right now. And that will give me a count or a concentration of E. coli in each of these samples. Then I'm going to put all 3 of the test tubes in a water bath for 2 hours. And then we're going to see how the samples compare 2 hours later. Now, if urine is antimicrobial, I expect the urine concentrations to go down. The nutrient broth concentrations should go up because E. coli has had 2 hours in really nice conditions for them, and so hopefully those concentrations will go up. And if azo actually does confer some antimicrobial activity on E. coli, we expect those concentrations to go down even more than the urine sample that was taken prior to consumption of azotabs. All right. Okay, so I have the first setup for my urine, not urine, this is going to be my control. So this is my E. coli with LB broth. And you'll recognize the setup. I have 3 bottles. I have a bottle that's labeled A, a bottle labeled B, and a bottle labeled C. In addition, I have, 4 Petri dishes that are labeled, this one is 10 to the minus 4 control. I have a 10 to the minus 5 control, a 10 to the minus 6 control, and a 10 to the minus 7 control. And what I'm going to do now is I'm going to run Lab 8 procedures as described exactly in Lab 8 right down to exactly how those dilutions are done. Okay, so I have my pour plates ready to go. I just need to add my agar. So I've taken it out of the incubator and let it cool out or cool off a little bit. And now I'm ready to make my pour plates. Typically you'd be getting some help with this, but I am all alone here, so making a little bit of a mess. Okay, so I'm going to let those harden. The test tube that has the E. coli and the the nutrient broth in it has gone into the water bath, 37-degree water bath, where it will be for 2 hours. And now what I'm going to do, and I'm not going to record this, but I'm going to do the same steps for these other 2 test tubes here. So these 2 test tubes are going to get the same treatment. I'm going to do serial dilutions and then make plates at the 10 to the minus 4, 10 to the minus 5, 10 to the minus 6, and 10 to the minus 7 dilution, and then go ahead and put those test tubes in the water bath for 2 hours. Okay, so it's been 2 hours and 20 minutes since I put the test tubes in the incubator. So I went ahead and grabbed them out of the water bath. And now what I'm going to do is I'm going to do another set of serial dilutions 2 hours and 20 minutes later for a total time of 140 minutes. So this one is the control. This is E. coli with the control. And you might notice that I don't have 4 plates set up here. I only have 3 of these plates. That's because I'm not going to do all of the dilutions that we did for the first set. Because I've got E. coli in nutrient broth, I am anticipating that a 10 to the minus 4 plate would still be too numerous to count. So I'm not even going to do that one to save on resources. Instead, I'm just going to do the 10 to the minus 5, 10 to the minus 6, and 10 to the minus 7. The urine ones, likewise, I'm going to adapt which dilutions I'm going to do based on what I expect to see, so that hopefully I can get a countable plate in each set. Hi, so it's the next day, and we are looking at the results of our pour plates. And so I have unstacked the pour plates, and I've started to take a look at them. And so what I'm going to do now is I'm going to actually just take photos of the pour plates, and I will go ahead and embed them into this video. You're now going to view 6 sets of plates. In each case, the plates will be shown to you as an image first, and then you'll see a video where I start using a Sharpie to try to help you identify the colonies. You'll need to follow along the Sharpie and count how many little circles are made on each plate. It's your job to record the number of countable colonies on each plate. If there are too many colonies, then what you'll see is that I'll write TNTC, which stands for too numerous to count. Good luck. There are clusters of colonies that are so tight that it would be very difficult to count. And this plate clearly has over 300 colonies, even though the colonies are very small.